Hello everyone. Good morning. How are you? Hope you all are quite okay and fine. I am also fine. Welcome you all to my today online class. Before going to start my today's lecture, first of all, I would like to introduce myself to all of you. My name is Mohammad Ghulam Morshed, Resignation Lecturer, Department Biology, Milestone College, Uttara Dhaka. Topics. Today I am going to discuss about some very important topics, that is the process of gametogenesis, spermatogenesis and oogenesis, differences between spermatogenesis and oogenesis, process of fertilization. These are the topics that will be discussed here today, and these topics are under the chapter 9 Human Physiology, Continuation of Human Life. Learning objectives. After completing my today's session, the student would able to know about, or you can define gametogenesis, spermatogenesis, oogenesis, acrosome, fertilization, vitelline membrane, zona pellucida, corona radiata. You can explain the process of spermatogenesis, oogenesis in detail. You can explain the morphology of sperm and ovum. You can explain the process of fertilization. To understand our topic very clearly, you should have some idea regarding what is gametes. Now, I would like to discuss about the gamete first of all. A mature male or female germ cell usually possessing a haploid chromosome set and capable of initiating formation of new diploid individual or new offspring by fusion with gamete of the opposite sex is known as gamete. So from this definition we can easily understand that the gamete is actually contain haploid set of chromosome, and when two opposite gametes are fused together to form a diploid individual or new offspring. Gametes are of mainly two types: male gametes, which is known as sperm, and the female gamete, which is known as ovum. Now let's discuss about what is gametogenesis. Gametogenesis term is actually derived from the Greek word gamos, which means marriage, and genesis means origin. So the gametogenesis is the process of the gamete formation in sexually reproducing animal. Sperms and the ova are produced from the germinal epithelium cells of the testes and ovaries of sexually matured male and female respectively through the process of gametogenesis. Now the definition of spermatogenesis and oogenesis. Spermatogenesis, the process of sperm production is known as spermatogenesis. The process of spermatogenesis occurs in the cells of the germinal epithelium of the male gonad or testis. Gonad is a simple terminology. What is gonad? Gonad is the primary sex organ that actually responsible for the production of sex hormone and the germ cell or reproductive cell. Gonads are mainly two types: male gonad, which is called the testis, and the female gonad, which is called the ovary. The oogenesis, the process of ova production is known as oogenesis. The process of oogenesis occurs in the cells of the germinal epithelium of female gonad or ovary. Now let's discuss about the process of spermatogenesis. To understand the process of spermatogenesis, clearly you should have some idea regarding the structure of the testis first of all. What is testis? As you know that there are two testes are actually located inside the scrotal sac in male body. The scrotal sac is actually lie outside the abdominal cavity in man. The testis is formed of about 1000 seminiferous tubules, and a special type of cell, leading cells, are also found in between the seminiferous tubules of the testis. The seminiferous tubules are long and coiled tubes lined with a single layer of germinal epithelium. The leading cell is actually responsible for production of testosterone hormone. The germinal epithelium cell consisting of two types of cell, Sertoli cell and the spermatogenic cell. Sertoli cells provide the nourishment to the developing spermatogenic cell, and spermatogenic cell give rise to the spermatogonia from which sperms are produced. This is the simple structure of the male testis or male gonad or testis, and this process of spermatogenesis process is actually occurs in the male gonad or testis. The human testis is composed of many seminiferous tubules which are lined by cells of the germinal epithelium cell, and this spermatogenesis process is actually completed 4 successive stages. Now let's discuss about the 4 stages very briefly. Number 1, the multiplication phase. The spermatogenesis is a continuous process. The germinal cells which produce the sperms are known as primary germinal cell or primordial germ cell. The cells multiply by repeated mitotic division and produce spermatogonium, which contain diploid number of chromosome or 2n number of chromosome. And second stage is called the growth phase. In this stage, spermatogonial cell accumulate large amount of nutrition to form primary spermatocyte, which also contain diploid number of chromosome or 2n number of chromosome. Remember that the spermatogonium cells and spermatocyte both contain 2n number of chromosome or diploid number of chromosome. And after that, at third stage is called the maturation phase. This is the actual meiotic phase. At first, each primary spermatocyte undergoes first meiotic cell division to produce 2 haploid cell, 2 haploid secondary male spermatocyte. Each secondary spermatocyte passes through the second meiotic division and produce 4 haploid spermatid. And as a result, after maturation phase, the diploid spermatogonium produce 4 haploid spermatid finally. And spermiogenesis, the metamorphosis of a spermatid into sperm through the complex process called spermiogenesis. Spermogenesis, in case of spermiogenesis, there are various changes occur in the spermatid. The first of all, actually, as you know that spermatid is a round shaped structure, but it has to be transformed into a elongated motile sperm. But there are some changes. There will be some changes in the spermatid. The first changes is that the nucleus of the spermatid shrink, discard water, RNA and nucleolus and form the head of the sperm. The second change is the Golgi apparatus is actually concentrated near the anterior end of the sperm nucleus to form acrosome. The mitochondria becomes spiral to form the middle part of the sperm, and the 2 centriole of the spermatid form the axial filament and the tail of the sperm. If you see the figure here, you can easily understand the spermatogenesis process. Look at the figure here. The spermatogonium is actually contain diploid number of chromosome or 2n number of chromosome, and this spermatogonium cell is produced from the primordial germ cells or primary germinal cells by the repeated mitotic cell division. So the spermatogonium cell is actually contain 2n number of chromosome or diploid number of chromosome, and this spermatogonial cell get accumulated large amount of nutrition to form a primary spermatocyte, which also contain the diploid number of chromosome. And after completion of primary spermatocyte, then meiotic cell division will be started. The primary spermatocyte undergoes first meiotic cell division to form secondary spermatocyte, 2 secondary spermatocyte which contain haploid number of chromosome. As you know that during the meiosis cell division, the number of chromosome will be half in the daughter cell. That's why the secondary spermatocyte contain haploid number of chromosome, that is n number of chromosome. And again the secondary spermatocyte again undergoes the second meiotic cell division, that is simply the mitotic cell division, to form 4 spermatid which also contain haploid number of chromosome. So from this figure you can easily understand the process of the spermatogenesis. Now let's discuss about the process of the oogenesis. The process of the oogenesis just similar to the process of the spermatogenesis. If you can understand the process of the spermatogenesis, then you can easily understand the process of the oogenesis. But there are some terminology will be different, but the process is just similar to the spermatogenesis process. The process of the oogenesis actually occurs in the cells of the germinal epithelium of the ovary. Such cells are known as the primary germinal cells or primordial germ cells. The oogenesis is completed in the following 4 successive stages. Number 1, the multiplication phases. The primary germinal cells multiply by the repeated mitotic cell division and produce numerous oogonia which contain diploid number of chromosome, 2n number of chromosome. And second phase is called the growth phase. In this phase, oogonia of the ovaries all get nutrition and become enormous, large in size. Egg cells in this stage are also known as primary oocyte which contain diploid number of chromosome or 2n number of chromosome. Third phase is called the maturation phase. This is the actual meiotic cell division. The maturation phase is accomplished by maturation or meiotic cell division. After the first meiotic division, the primary oocyte divides unequally to produce a large size haploid secondary oocyte which contain haploid number of chromosome or n number of chromosome and a small size haploid first polar body. The secondary oocyte passes through the second meiotic division to form a large matured and a small polar body. By the second meiotic division, the first polar body also divides into 2 secondary polar body. So, thus by the maturation phases, a diploid primary oocyte produces ultimately 1 large haploid oocyte and 3 small haploid polar bodies. Finally, now the metamorphosis process. In this phase, the oocyte modified into a mature ovum. But in this case, no huge changes as like in spermatid. The polar bodies ooze out from the egg and degenerated while haploid ovum ready for fertilization. Please see the figure here. You can easily understand. If you see the figure here, then you can easily understand the process of the oogenesis. At the top of the figure, the oogonium. Oogonium cells contain diploid number of chromosome, that is 2n number of chromosome. The oogonium cell is actually produced from the primordial germ cell or primary germ cell by the repeated mitotic cell division. And primordial germ cells is also contain the diploid number of chromosome. As you know that during the mitosis cell division, the number of chromosome remains same in the daughter cell. That's why the oogonium contained a 2n number of diploid number of chromosome, as same as the germinal epithelial cell or primordial germ cell. And this oogonium, during the growth phase, the oogonium get more nutrition and becomes enormous, large in size, which is called the primary oocyte. And the primary oocyte is actually contain 2n number of chromosome or diploid number of chromosome. So remember that the oogonium and primary oocyte both contain diploid number of chromosome. And after that, the primary oocyte undergoes first meiotic cell division, this is meiosis 1, to produce a large size secondary oocyte and a very small size primary polar body. And secondary oocyte again undergoes second meiotic division to produce a large size oocyte and a small size polar bodies. Similarly, the primary polar body undergoes second meiotic cell division to produce two very small size secondary polar body. And ultimately, after maturation phases, each primary oocyte ultimately produce a single large size oocyte and the three small size polar bodies. And this single large size oocytes are ultimately transformed into the ovum by the metamorphosis process phase. Now let's discuss about the differences between oogenesis and spermatogenesis. Oogenesis actually occurs in the ovaries of the female. Spermatogenesis occurs in the testes of the male. In case of oogenesis, one gamete produces per meiosis, but in case of spermatogenesis, 4 gametes produce per meiosis. In oogenesis, completion of the meiosis is delayed for months or years. But in spermatogenesis process, the meiosis is completed in days or weeks. In case of oogenesis, fixed amount of ovum produced throughout a female life. In case of spermatogenesis, billion of sperm produced throughout a male life. In case of oogenesis, fertilization stops at menopause. But in case of spermatogenesis, fertility is lifelong but reduced in age. In case of oogenesis, gamete release in monthly cycle, but in spermatogenesis process, the gamete release in any time. Now let's discuss about the morphology of the sperm. Each human sperm is 50 micron long and consists of head, neck and tail. Head is the first part or anterior part of the sperm. The anterior elliptical or spherical part of the sperm is called the head. It has a cap-like acrosome that secretes hyaluronidase enzymes that helps in fertilization. Neck. Next is the second part of the sperm. This is the very constricted, narrow and short part just behind the head. It contains centrioles, which are proximal centriole and distal centriole. And tail is the last part of the sperm. The neck follow a long, narrow part called tail of the sperm. Distal centriole give rise to axial filament of the sperm, which run up to the end of the tail. If you see the figure of the human sperm, you can easily describe the structure of the or morphology of the sperm clearly. Now the morphology of the ovum. The female gametes of sexually reproducing animals are called ova. Human ova are extremely minute, measuring about 0.2 mm. Human ovum consisting of following structure. Covering the ovum is enclosed with 3 covering, vitelline membrane. All ova are covered with the cell membrane or plasma membrane called vitelline membrane. Primary membrane. This membrane formed when ova are in ovary. In mammal, it is designated as zona pellucida. See the secondary membrane. It is as additional membrane formed when egg pass through the oviduct. In case of mammals, this layer is known as corona radiata. If you look at the figure here, you can easily understand that the ovum is actually enclosed by 3 layers. The innermost layer is called the vitelline membrane, and the middle layer is called the zona pellucida, and the outermost layer is called the corona radiata. So the ovum is actually encircled by the 3 layers, and the ooplasm and the deuteroplasm. The cytoplasm of the ovum is called the ooplasm. In most cases, ooplasm contains reserve food as yolk or deuteroplasm, which determine the size of the egg. Nucleus. Nucleus of the ovum is round in shape. It has haploid number of chromosomes. Fertilization. Look at the figure here. This is the process of the fertilization. There is a ovum and so many sperms around a ovum, and trying to penetrate inside the ovum. Fertilization is a process by which haploid male gametes and female haploid gametes fuse to produce diploid zygote. Fertilization is a special biological process which activates the ovum for further development. Fertilization is species specific. Sperm and the ovum of the same species can fuse. Now let's discuss about the process of the fertilization. When sperm come nearer to the ovum, fertilizing chemical and anti-fertilizing chemical secreted from ovum and sperm respectively. Due to the chemical interaction between fertilizing and anti-fertilizing, attachment of sperm to the ovum takes place. This process is called the agglutination. Secondary oocyte is surrounded by zona pellucida and corona radiata. When sperm remain close contact with the wall of the ovum, special enzyme hyaluronidase is secreted by the sperm, which dissolve some cells of the corona radiata, making a penetration pathway for sperm into the ovum. Then another enzyme, namely acrosine, secreted from sperm, which helps sperm to occupying it in between membrane of the ovum and the zona pellucida. Now fusion takes place between plasma membrane of the secondary oocyte and sperm. Then sperm penetrates inside the ovum. The secondary oocyte undergoes second meiotic division to form a single ovum and 3 polar bodies. Now fusion of pronucleus of sperm and the pronucleus of ovum take places and form diploid zygote. This is the process of the fertilization. This is the figure of the fertilization process. Now the homework, write down the differences in between spermatogenesis and oogenesis. Creative questions. Dr. Mohsen said in a seminar, there are many types of cell in human body. Among them a special cell type of cell which is different in shape and they are produced in male and female body. Human life cannot be imagined without this type of cell. A. What is spermatogenesis? B. What is meant by oogenesis? C. Explain the special process of producing of mentioned cell in the female. D. Give a logical opinion about the last sentence of this team. Thank you all for your patient sharing. I think today's lecture is a very effective lecture for all of you. You all are requested to watch my video lecture for several times so that you can understand each and every term of my today's lecture very clearly. If you have any queries regarding my today's lecture, feel free to ask me. You can ask me anytime, or you can ask your respective subject teacher regarding your queries. We all are prepared to solve your problem. Thanks for all. Thanks you everybody.